Okay, so well, glad to be here live to present you an update on where we stand with the development of Leriglitazone. I will try to keep it simple, although you will see some technical aspects in the slides. And I'm very enthusiastic always with our result because I'm very confident about the drug, but take it with caution, my comments, because at the end of the day, we are still in development and with some work still to be done to confirm all what we are seeing. Having said that, let me introduce the company in a single slide, just for those of you that don't know us. We are a little biotech from Barcelona that I founded now 10 years ago with the ambition to develop drugs in the orphan CNS space. And today we are essentially focused on Leriglitazone. Is a small molecule, is an oral treatment that allows to exploit a biology called PPAR gamma agonism in the CNS. This is an approach that has quite a lot of potential in CNS, as I will describe later. But what has been lacking in the space is a PPAR gamma agonist able to reach the brain in sufficient levels in humans. And that's precisely what we have, and which is an approach that have a potential in several indications. In our case, our lead indication and where we spend most of our time is in XALD, in Adrenoleukodystrophy, both for Adrenomyeloneuropathy and the cerebral form of the disease. And that's essentially what I'm going to be covering today, our efforts in ALD. We are located in the north of Barcelona, but also we have a smaller team in Belgium. And before moving into the science and into an update, just let me show you who is supporting the company, and also as an opportunity to thank them for their support. We have a pretty large network of investors, European investors, supporting us from Belgium, Spain, France, Switzerland, etc. And also we have quite a lot of support from different institutions around Europe, and this includes European Union itself, but also the Spanish government and also the Belgian government and also the Catalan one. So overall, many thanks to all of them, and also to our partner in China. We already licensed the molecule for the Chinese market, and that's our partner Spirogenics that we are working with. Okay, that was more the boring part, if you want. So I guess that the real interest here for today is where we stand with the molecule. Leriglitazone, as I said, is a small molecule that acts on PPAR gamma agonism. The reason why this has a potential in CNS is because PPAR gamma agonism has been involved into activities such as neuroinflammation, myelination, neuronal death, and mitochondrial function. And these are drug class that the risk benefit is pretty well understood, because there's been already drugs in the market targeting PPAR gamma agonism for other conditions non-CNS related. And one of these drugs is pioglitazone, or Actos, the commercial name. Actos has been used for treating diabetes, and there's been hundreds of thousands of patients being treated with a PPAR gamma agonist for the treatment of diabetes. So this is the safety profile is pretty well understood. The issue with pioglitazone or any other PPAR gamma that's been used in the past is that they do not reach the brain at sufficient levels. As you know, for the development of a brain treatment, we have two key issues. One is the usual safety and efficacy component, as any other drug, but the second is that we need to be able to cross the blood-brain barrier at sufficient extent to reach the target, which is in this case the CNS. Our molecule, actually, we talked about this in the past here, but is a metabolite of pioglitazone. So it's a molecule related with pioglitazone, but this specific metabolite, among the many that pioglitazone has, this specific one has a superior profile for CNS, and that's for different reasons: pharmacokinetics, brain penetration, safety profile, metabolic profile. So there's a number of different reasons. So without entering into all the tiny details here, this molecule allows to reach in the CNS the right levels of the molecule to be able to be efficacious in CNS disorders such as ALD. And that's something that we already shown in phase 1 clinical studies long time ago. We are talking about data from 2015, 2016. In there, we were showing that the molecule really crosses the blood-brain barrier and engages the receptor at the right level, compared with what we got in preclinical models. And this molecule is orally bioavailable and is formulated as an oral suspension. Maybe, I don't know, but maybe some of you have been involved in one of the trials that we have performed, and you know that this, the drug comes in a bottle with the syringe, so similar to the ibuprofen for kids, to just give an example. But and that's a pretty simple process because it's once a day administration after breakfast or before breakfast, so it doesn't really matter much. So it's pretty easy from that perspective. The reason why we are doing this as an oral suspension instead of the typical tablet is for multiple reasons. One is because this allows us to make sure that all patients reach the right level of the molecule. So on a patient-by-patient basis, we can adjust the dose after a few days of treatment to make sure that all of them are at the same level. But this formulation also allows the pediatric development, which is very important for us, as you will see later in the studies that we are conducting. Finally, in ALD, but also in other indications that we are considering, sometimes when patients get more progress, there may be problems such as swallowing difficulties, to give an example, and this kind of formulation also offers a broader flexibility on this regard. We have data in many different conditions, not just ALD. As I said, ALD is our primary focus, and most of the data is around ALD related, but we also have data in other conditions such as Leriglitazone ataxia, where we also conducted a clinical proof of concept clinical study that I'm not going to comment today, but also other important CNS disorders in the orphan space, mostly. CMC, what refers to the manufacturing process of the molecule. This is already industrial phase, so getting ready to move to the, hopefully pretty soon, into the commercial phase. And the molecule, we also got orphan drug designation, both in Europe and also in the US for ALD, also Leriglitazone ataxia. And for the case of ALD, in the US, we also have fast track designation and rare pediatric designation. This is about how the molecule works, and I'm not going to enter into all the details here, just to make us a complex story short. We published this in the Science Translational Medicine recently, so it's a very nice publication. For those of you with interest to go deep in the science, in this publication, all the details are there. Essentially, the idea here is that ALD is a simple disease in terms of the genetic basis. So it's a monogenic condition. So it's a single gene that is affected. In this case, as you know, ABCD1, that is in charge of transporting very long chain fatty acids into the peroxisome. And that's easy to understand if you want. The problem is that down the road, downstream, ALD becomes very complex. So there's multiple pathways that become impacted in patients, and is not as an easy disease at all. So the beauty here is that PPAR gamma is a transcription factor that is controlling multiple pathways, multiple aspects in the cell that are very relevant in the NCNS. And in this case, what happens is that with this specific mechanism, we can modulate at the same time these multiple pathways that are relevant in ALD. And this goes from inflammation through blocking NF-kappa beta, also to neuronal survival, remyelination, is a demyelinating disorder in this case, and also importantly, a lot of control on mitochondrial function, which is also central, particularly in AMN in this case. This slide is essentially the mechanistic part for any ALD case, but then we also have the situation that some patients may develop cerebral form of the disease, which is we don't understand that well at the molecular level why some patients develop cerebral ALD on top of the possibility of AMN. And what we have seen here in multiple models is that the drug, Leriglitazone, may have an effect on different steps that are key for cerebral progression. And this goes from the initial disruption of the blood-brain barrier to the activation of microglia, astrocyte survival, neuronal death, and myelination. So the data supports that this molecule, leriglitazone, may have potential both for the chronic AMN form and the acute cerebral ALD form. Now, a long time ago, because this is something that we started in 2017, we moved in, as a first step in the space into a trial, a phase 2/3 trial with the ambition to become a registration-enabling study in AMN. That study that we discussed at length with QLs, patient advocates, FDA, EMA, and this was a study that was focused on AMN, men with AMN. In the slide, you have the different sites that have been involved, and I'm sure you recognize the names. In UK, we work with Robin Lachman. In London, but as you can see, we work really with all the reference sites across Europe and in the US. Actually, this is the largest trial ever in AMN so far. And it was a study that was 2 years treatment, double-blind, placebo-control, and 2-to-1 randomization, meaning that for every 2 active, there was 1 patient receiving placebo. And we randomized more than 100 patients. Most of them completed the study, and a significant proportion of them moved to the open label extension that continues today. Going to the results of the study, that's what probably most of you are familiar with. Unfortunately, the primary endpoint, which was a measurement of the 6-minute walk test that is related with the how long can you or how far can you walk in 6 minutes, this was not met in the entire population. At the same time, the key secondary, which was a measure of balance or body sway, in that measure, we were able to see in multiple sway-related secondary endpoints, we were able to see very nice efficacy. Body sway is a measure of balance, as I said, and as you know, balance is an element that is largely impacted in AMN because of the degeneration of the dorsal column. So these results were suggesting that even though we were not able to see an impact on the 6-minute walk test, the drug may be having a benefit on myelopathy, and that is seen or observed in the impact on the balance. At the same time, one another important aspect in our trial was the assessment if these adult AMN patients were developing cerebral progression or not. Because as you know, cerebral ALD, even if it's typically seen in pediatric patients, it's also pretty frequent in adults with AMN over time. Actually, the most recent literature suggests that up to 60% of AMN patients may develop cerebral ALD over their lifetime. And at that point, it becomes as bad as is the case for pediatric patients, unfortunately. In the study, we used central reading of all the imaging data. So it was centralized by the same group of people looking at all the MRIs from all the different sites from all the patients. And there we could see a significant reduction of the progression of cerebral lesions in the brain in treated patients compared with placebo. At the same time, when we take into account the clinical picture, clinically, only placebo patients in the study developed cerebral ALD. So a total of 6 out of 39 for known out of 77 in the active. So suggesting that there's a significant reduction of developing clinically CALD. And that was well aligned with another secondary endpoint, in this case, the Loess score that had a larger increase in the placebo compared with the active, and also multiple biomarkers, neurofilament light, MMP9, which has to do with blood-brain barrier integrity, interleukins that have to do with inflammation. So all of them providing a consistent picture that the drug is working on cerebral progression in line with the mechanism of action and preclinical data. On top of this data that for us is very encouraging, we were able to see strong trends in clinical rating instruments in the entire population, so EDSS, SSPROM, even global impressions, and getting very close to this level of p-values also in the quality of life. So overall, we think that really the trial is providing a global picture that the drug is having a meaningful effect on AMN patients, both in terms of myelopathy progression, but also importantly on the cerebral progression. The question remains of what about the primary, why we missed the primary endpoint. And on that, we did some post-hoc exercise. So meaning that being post-hoc, this needs to be taken with extra caution. But in this post-hoc exercise, what we were able to see is that ambulation-related endpoints, and here includes 6-minute walk test and EDSS ambulation, which is another endpoint of the study. In ambulation, we do not see a linear decline in AMN patients. What we see is that there's an initial phase where there's a steeper decline, so closer to the onset of the disease, followed by a second phase where the decline in the ambulation is much lower over time, at least in 2 years time frame. And what we observe also is that in this group of patients in the early phase of the disease, in the phase where the decline in ambulation is steeper, in these patients we do see a treatment effect both in 6-minute walk test and ambulation, and EDSS ambulation. So suggesting that maybe the lack of effect in the entire population in 6-minute walk test is coming from this issue between populations. But again, this is a very reassuring or encouraging if you want, but being post-hoc, it needs to be taken with extra caution, as I said. In terms of safety, the drug was generally well tolerated. So it's not an innocuous drug as any, I would say, but the most common adverse events were weight gain, which can be in the range of 5, 6, 7, 8% in this range in general, but it reaches a plateau really, so it gets stabilized at one point. Edema is also pretty frequent, but again, is in general manageable and moderate for most of the patients at least. So overall, the benefit risk profile for the molecule looks pretty good, considering the disease that we are discussing. With that results, we went to see regulators, and this is where we are now. And the discussion with regulators, we already had discussions with the rapporteur team appointed by the EMA in a pre-submission meeting. So essentially, after this meeting, we took the decision to move into filing a marketing authorization application in Europe. Something that is going to happen, hopefully, if nothing goes wrong and everything is on time, during the second quarter next year in 2022. And will be followed by a UK filing as well, if everything goes smoothly in that process. In the US, we also had discussion with FDA. Initially, maybe some of you are aware of those discussions, but initially with FDA, because of the lack of efficacy in the primary endpoint in the entire population, FDA had the initial impression that it was a failed study, and that put under question the global benefit risk profile. We provided more data to FDA, particularly on all these results around cerebral lesion progression, also at the body sway, but essentially on the cerebral lesion progression. And based on that data, FDA is allowing us to continue with the treatment of open label extension patients in the US. So at the same time, FDA showed significant interest on all these new results. So we are having additional discussions with FDA to define how we can generate additional evidence to allow us to move to an NDA. What is clear is that with what we have now, this is not going to be sufficient, and we are going to need more evidence for the FDA, and that we are going to be working on that. At the same time, we're also working to extend the use of Leriglitazone beyond Europe and in the US. So we are having already an agreement, or we have an agreement already in China, but we are working also to expand this into other regions around the world. That's about AMN. Let me also provide a brief update on where we stand in CALD. In here, we have an ongoing study in pediatric patients. As you know, in CALD, there's an initial phase where the first aspect of the disease is the appearance of lesions in the brain. After that, the clinical symptoms appear, and after that, patients move into pretty acute progression in average time of 3 years, leading to death or vegetative state, unfortunately. The transplant-based therapies, either the hematopoietic stem cell transplant or ex vivo gene therapy, are performed after the appearance of brain lesions. And we are now using leriglitazone at the very early start of the disease with the goal to see if we can avoid or delay the need for a transplant-based process for these patients. And that's where we are placing leriglitazone at this moment. This is an ongoing study called Nexus. This is targeting or treating boys between 2 and 12 years with lowest score up to 10. It's an open label study. And what we do is we treat the patients and we see if we can achieve stabilization or deceleration of these lesions in the brain. If that's the case at 6 months, then treatment continues. And important to highlight that this is not interfering with hematopoietic stem cell transplant if it becomes indicated. At any point of the study, patients can go to transplant if it becomes needed. So we do not interfere in that. This is a study that is recruiting. So if you are aware of patients that could be interested in this, of course, happy to consider if they could be eligible. All the traveling costs are covered to the sites that we are having in Spain, France, or Germany. And we even had already patients in the study coming from as far as Chile. So all that is covered. So that's not an issue. You have the sites or the PIs of the study in the slide. Also, you can recognize for sure the names. And the principal investigator of this study is Patricia Mussolino from Mass General in Boston. That is also deeply involved in the design, also and also the conduct of the study, because we centralize all the imaging data in there as well. That's it for the presentation. I hope that everything was clear, but I'm happy to answer any question that you may have.